So we're the SciStarter team. SciStarter is actually a non-profit run by a friend of mine, Darlene Cavalier. I'm actually not part of SciStarter. I'm collaborating with SciStarter. I'm a microbiologist at the University of California, Davis. So I'm in the wrong time zone. We have a pretty small team. Me, Mark, and Camden and Anne were here yesterday, and they seem to have gone away. Anyway, together with SciStarter, UC Davis is doing a project to actually run an experiment on the International Space Station. We won a contest by a NASA contractor called Nanovax. So we're actually sending a little 4x4 experiment to the space station. We're going to be doing a microbial growth experiment on their microplate reader. And this is part of a larger piece of research sponsored by the Alfred P. Sloan Foundation on microbiology of the built environment. And the reason why this is so interesting to me is that I study microbes in the environment, like volcanoes and hot springs. Next year I might be going to Antarctica, hopefully. It turns out we actually know more about the microbes that live out in the environment in weird places like the deep sea than we do about microbes that live in buildings. And so the Sloan Foundation has been very interested in trying to encourage more building scientists and microbiologists to actually study these things. So as one of the things that we do in microbiology or microbial ecology is we like to look at large data sets that compare lots and lots of different places, like hundreds of different sites with thousands or tens of thousands, hundreds of thousands of different taxa, all the genes that we know about. For example, we have a project that in our laboratory where we've been trying to just figure out how many different kinds of proteins are there, and our current estimate is on the order of 500,000. So, and running. This is what our data looks like. This is actually a reanalysis of some older data from the Global Ocean Survey, which Craig Hunter did. And in microbial ecology, we always end up making these heat maps, which are sort of slices of these big multivariate databases. We have to take two different variables and splay them out across two axes and cluster the numerical data. And making these heat maps is really important for interpreting the data that we get. But the tools that we use to make them are sort of clunky. I don't know how many of you are familiar with R, but it's a very powerful statistics package that takes years and years to learn how to use it properly. And we're doing a citizen science project for the space station sampling project. We're also sampling, sports arenas and things like that. We really want to be able to involve people in the public in data analysis and interpretation. So we thought it was really important to be able to actually make these heat maps and play around with them and be able to do your own, for people participating in the project, to be able to actually interpret the data that we're generating on the fly, since we're posting all of our data publicly. To avoid having to dig around in R, we thought it would be really awesome to be able to make heat maps from the raw data yourself. So we built a tool to do that. It's a little bit, I'm not a graphic designer, so it's not actually very pretty, but it does work. This is raw data from the Global Ocean Survey. Basically, sites in the ocean go this way, and proteins go this way. And these are the co-occurrence matrices. So that's the, so you can see we actually truncated this at 800. It goes way, way down, because it goes to all the proteins in the protein family database. So it's a little bit better design. We could make these pixels a little smaller this way, and a little bigger this way, and fit it into the screen. And so the idea is, I don't know if you guys heard of the ENCODE project. There was a big announcement about it a little while ago. In their Nature paper, they had this figure, which is what I was thinking about when I decided this would be a really awesome visualization tool. So this is what the data looks like if you go on here. These are human genes, and this is a co-expression matrix, right? And so this is what it looks like if you sort the data alphabetically. It's not really it just looks like a mess, right? And so they made this thing so you can recluster it, and you can so that you can see which genes are actually expressed at the same time. So these little clusters represent genes that are expressed simultaneously with this data set. And like I said, it would be really awesome to do with our tool. And when we started working on it, it's not quite done yet. We only have one clustering algorithm working. But to be able to basically pick which clustering algorithm you want to use, right? And then what cutoffs and what other parameters you want to use. So you can actually do, the sort of the statistical analysis and the interpretation part of these large-scale microbial experiments. So.
Thank you. I think maybe the team that came the furthest to come attend the Philadelphia.
Yes.
So please welcome our guests and thank them.
If you go to the Phillies game later this week, we're actually going to be collecting samples at the game.